Excellent. Hi, everybody. Thank you for joining us. I'd like to welcome you to the Plant Ecophysiology webinar, focusing on a topic that's published this month in Plant Physiology. And we have three speakers today, Amanda, Thomas, and Sabina. And our webinar is hosted by one of the editors of this special focus collection, Agustin, fill in the name because I can't remember how he told me how to say it, but it's brilliant when he would tell us. The webinar is moderated today by one of the assistant features editors, Thomas de Papa, and I will introduce him in just a second. I'm just going to run through a few housekeeping slides. There we go. If you look at the Plant Physiology website, you'll see this beautiful cover that Augustin contributed, really trying to capture some of the topics that is covered in this focus collection. And after the webinar, we'll be posting this to YouTube. And if you go to the YouTube Plant Physiology channel, you'll see all of our past webinars on every topic we've covered are available there. I would like to thank the ASPB members who are joining us, because the membership of the society helps support programs like this. And if you decide to join as a member, you use the code presents10 and you get 10% off your membership dues. I'd also like to remind you that this year the Plant Biology the Plant Biology Conference is held in conjunction with the Canadian Society of Plant Biology. So, oh no, that's last year's. Oops, I should have updated my slides. So this year it's in Ottawa, Canada. I'll put the link into the chat in a few minutes. We'll just skip this slide. If you have any questions for the speakers, please put them into the Q&A box at the bottom of your screen. You can email me if you have any problems. I will be sending out a certificate of attendance, but in order for you to receive it, you'll receive an email in 24 hours after the live event. You have to follow the instructions on the email, and then you will automatically get your certificate of attendance. So now I'm going to introduce our moderator, Thomas. He received his PhD from Ghent University in Belgium, and he carried out his postdoctoral work there as well. He was investigating the physiological roles of hormones that regulate growth and stress adaptation in Arabidopsis. And then he moved to Spain and joined the Plant Synergy group at the University of Barcelona. He's currently an assistant professor at the University of Barcelona, and his research interests are in organelle communication, combined abiotic stress, and hormone signaling. So I will turn things over to you, Thomas. Thank you.
Thank you very much, Mary, for the kind introduction. I'm delighted to be here. But let us go ahead and kick off the seminar by introducing the host, Professor Agustin Zsögön, if I'm pronouncing correctly. Agustin is an assistant professor of molecular plant physiology at the Federal University of Viçosa, Brazil. He is both a geneticist and a plant physiologist, and focuses on gene discovery for the de novo domestication of crop wild relatives, with the idea of finding targets for increasing abiotic stress tolerance. So at the heart of his work lies ecophysiology, which is to understand how plants function in real environments and how genetic changes translate into physiological performance under conditions such as drought, heat, and other stresses. Reflecting his engagement in this field, Agustin, together with 5 colleagues, guest edited a focus collection dedicated to this important area of plant physiology. So, Agustin, the floor is yours.
Thanks, Tom. Thank you very much for the nice introduction. So, hello again. Good morning or good afternoon, depending on your time zone. It's my huge pleasure to host the first webinar of the year, and it's a special year because it marks the 100th anniversary of plant physiology. And just by coincidence, it is also the 100th anniversary of our university here, the Federal University of Viçosa, where I work, as Tom said, and where also Professor Wagner Araújo works, who did a lot of the heavy lifting to edit this focus collection on plant ecophysiology, along with our co-editors, Scoffoni, Tracy Lawson, Florian Busch, and Xinguang Zhu. So we are very excited and happy to wrap up this very nice collection of articles with this webinar today. 25 research papers, 7 update reviews, and a couple of news and views. We received lots of submissions from all over the world, so we really enjoyed putting together this special issue to provide an overview of the state of the art on plant ecophysiology. So plant ecophysiology is an experimental science that deals with how plants respond to their physical, chemical, and biological environment. In other words, it addresses what appears to be a simple question: how do plants function in real, dynamic, and often stressful environments? Answering this question requires integrating processes across physical scales, from molecules and cells to organs, whole plants, populations, and ecosystems, and also timescales, from seconds, or sometimes even less, nanoseconds, picoseconds, to years and decades. Ecophysiological studies have significantly enhanced our understanding of photosynthesis, respiration, water relations, plant nutrition, and plant responses to biotic and abiotic stresses. So this focus collection that we put together brings current mechanistic insights into fundamental ecophysiological questions by combining advanced and emerging approaches related to the study of plant function in general. So let me briefly highlight the contributions by our speakers today, who will be properly introduced by Thomas in a few minutes. So without spoiling too much, Amanda and co-authors provided a comprehensive review on plant hydraulic traits and crop performance under water limitation. They summarized the current understanding of how hydraulic conductance, stomatal traits, and related mechanisms influence crop yield during drought. Tom and his co-authors explored how genome-scale metabolic models can be constructed and used to predict metabolic responses to environmental conditions like elevated CO2, drought, and high temperature. And last but not least, Sabina contributed an original research article investigating stress tolerance in the drought-resilient houseleek. And they showed that under prolonged drought, this species employs a quiescent-like survival strategy, shedding light on inherent drought-resilient strategies in perennial species. So collectively, this collection challenges us to rethink where and how key processes occur. For example, the contributions move beyond the leaf-centric view of plant function, highlighting the roles of non-foliar tissues, whole plant integration, source sink dynamics, and so on. Many of the studies combined field experiments with high-resolution measurements, imaging, molecular analysis, and modeling approaches. So this combination allows us to move from correlation toward causation, and therefore to move forward from description toward prediction. So I guess today's webinar is a great opportunity to hear from these authors who have produced such outstanding research and explore some of these themes in more depth. We hope that this discussion will be engaging and appealing to you all. And thanks again to Mary, thanks again to Plant Physiology for this amazing opportunity. Thank you very much all for joining us, and please enjoy. With that, let's begin. And on to you, Thomas, again. Thank you very much.
Thank you, Agustin, for the overview of the special issue and of the webinar of today. I'm sure it will be a very interesting series of talks. So I will be brief to let the speakers have their time for the presentation. So I will introduce our first speaker of today, which is Professor Amanda Cardoso. Amanda obtained her PhD at the University of Tasmania in Australia, where she worked under the guidance of Tim Brodribb, where she studied the different aspects of drought physiology, focusing on both stomatal responses and xylem functioning. She then moved to the United States, first for a postdoctoral stay, and later incorporated in the North Carolina State University as an assistant professor. There she's leading currently a research group on ecophysiology of woody and herbaceous crops. And the talk of today is also centered around the work she carries out in her group. So today, Amanda will present a talk titled Plant Hydraulic Traits Influencing Crop Production in Water-Limited Environments. So in this presentation, she will explore how traits such as stomatal conductance, hydraulic conductance of leaves and roots, and related hydraulic characteristics can influence crop yield under changing environmental conditions. So with this, I give the floor to you, Amanda.
Thank you, Tom, for this nice introduction. Just a brief correction, I gained my PhD from University of Viçosa, but I was a visiting scholar in Tasmania for a year during my PhD. But can you all see my screen? Yes?
Yeah.
Thank you. Okay. So thank you for this introduction, Tom, again, thank you. Also the ASPB people for inviting me to be here presenting today, highlighting my work. I'm looking for the pointer, sorry. Okay, there we go. So I am very excited to describe this review that I wrote with some colleagues. And the review aimed to understand the influence of plant hydraulic traits into crop production in environments that suffer with water limitation. What I mean by plant hydraulic traits is any structural or physiological trait that influence the water movement throughout the soil-plant atmosphere continuum. In here, we focus on the stomata, which governs water loss from the leaf to the atmosphere, and also on the hydraulic conductances or the efficiency with which water moves along the plant, which directly influences water uptake and transport throughout the plant body. Now, the importance of water movement throughout the plants becomes clear when we see the direct relationship between evapotranspiration or crop water use with yield. So in environments where plants do not experience water limitation, we can expect genotypes that use more water to also produce more. Now, this is not exactly the case for environments prone to drought. So drought can hit throughout any time of the season. Plants can experience drought at different developmental stages. The most critical ones tend to be the reproductive ones. And for instance, in maize, the developmental stages as VT and R1, which represent the developmental stages at which male and female flower are developing, they tend to be the most susceptible ones to production losses when plants experience drought. And the result is what we see in here. Kernels or grains that do not set, develop, or fill. Now, there's not much a plant can do when drought hits them at those critical reproductive stages. Yield losses are going to occur. Now, what a plant can do is to have strategies to use water smart and efficiently, and even prevent excessive water loss when needed, such that water is conserved in the soil, and that minimizes the chances of drought to develop during those reproductive stages. And this strategy is what is described by the limited transpiration theory, and this has been demonstrated to be effective in mitigating production losses in environments prone to terminal droughts. Two components that will contribute to this limited transpiration strategy are improvements in water use efficiency and drought avoidance. So we describe a lot those two components in this review, and we talk about what hydraulic traits that influence and result in greater water use efficiency and in improved drought avoidance. I would like to highlight first a few hydraulic traits that influence the water use efficiency, and moderate reductions in the daytime stomatal conductance without major declines in photosynthesis appears to be the most cost-effective way to improve water use efficiency. Of course, we can also increase water use efficiency by increasing photosynthesis, but decreasing stomatocondutance seems to be more effective. So this is only possible because of the non-linear relationship between stomatocondutance and photosynthesis. We can, for instance, decline partially or moderately stomatocondutance without experiencing much declines in photosynthesis, and that will cause a huge increase in water use efficiency. Two hydraulic traits that are directly related to this. One is, of course, stomatal form and function. We can cite declines in stomatal density, for instance, and also decreased plant hydraulic conductances, which will reduce the supply of water to the leaves and make the stomata to be partially closed. Now, in another review for this special issue, me and some other colleagues describe in detail a number of recent attempts to decline stomatal density or genetically engineer plants to decrease stomatal density in the hopes of decreasing stomatal conductance and not have any negative impact in photosynthesis. I can briefly say that the most successful cases involved C3 and C4 monocots and barley, rice, and sorghum, and they were able to partially reduce stomatal conductance with no to low reductions in photosynthesis and yield. If you are interested, you can also check out that other review where we talk about this in more detail. Now, talking specifically about drought avoidance, increase the stomatal sensitivity to soil dehydration is a very effective way to improve drought avoidance. So when water lacks in the soil, plants that perceive that faster and trigger a stomatal closure to decline water loss are the ones that will be more benefited by this environment. So this is one of the ways that people have been using to identify the stomatal sensitivity to soil dehydration, which is essentially evaluate transpiration at the whole plant level as a soil declines in water. So as we decline, we decrease the fraction of transpirable soil or water. And the plotting number A shows two different contrasting genotypes of maize with different abilities to perceive and respond to soil dehydration. We see hybrid 17 in orange, declining transpiration at soils with more water availability, so early during a drought, than the hybrid in blue. So this hybrid would be more, have a higher sensitivity to soil dehydration and would be more benefited or prevent more efficiently declines in production in environments prone to drought. Now, beyond this natural variation in the sensitivity of stomata to soil dehydration, an interesting approach has been to change plant nutrition to also induce this phenotyping. So there we highlight the study that used two sources of phosphorus fertilizer, triple superphosphate and diammonium phosphate, and using the same genotype, and they observed that the use of a particular phosphorus fertilizer source also resulted in that phenotype where plants will start to trigger declines in transpiration earlier during a drought, so being more conservative in their water use. Now, the specific hydraulic traits that are associated with this phenotype are usually the ones associated with roots, where the soil drought is going to be first perceived. And some root traits that are associated with this phenotype are roots that rapidly lose physical contact with the soil, and also roots that experience earlier declines in hydraulic conductances as soils dehydrate. There are also some studies that show declines that leaves with lower stomatal conductance also can have this phenotype, but there's more discussion about that. Now, another component that is very important of drought avoidance is the increased stomatal sensitivity to dry air or high vapor pressure deficit or VPD. So in those conditions, plants will tend to lose more water for the same amount of stomatal aperture, and that will naturally decline water use efficiency in a way that plants cannot control very well. What plants can do is to minimize water loss under those conditions of high evaporative demand and be more sensitive to these changes in air dryness. In here, we also show some data of 2 different soybean genotypes that have different sensitivities to increases in air VPD, with the genotype presented in orange being the one able to more efficiently limit the transpiration as VPD increases, and the genotype in blue with a less sensitivity of the stomata to increases in VPD, such that leaf transpiration continues to increase with the increases in evaporative demand. Now, hydraulic traits that have been associated with a more efficient stomatal sensitivity to VPD are plants with low hydraulic conductances. That can be in the leaves or in the roots, and both of them seem to work very well and lead to plants that better sense dry air dryness. So just to conclude this brief presentation, plant hydraulic traits have important implications for both water use efficiency and drought avoidance, which in turn are very important for production in environments prone to water limitation. Engineering crops to reduce stomatal density and conductance holds promise for improving water use efficiency with low to no yield penalty. Of course, this response remains to be confirmed at the field level. And low hydraulic conductances result in greater stomatal sensitivity to soil and atmospheric drought, which are critical for avoidance and linked to production success in water-limited environments. If you want to see the full paper, we have the QR code in here. And with that, I would like to thank the collaborators that helped me to prepare this review. Thank you.
Thank you very much, Amanda, for a very interesting view on drought responses in plants and crops in general. So I think we have time for 1 or 2 questions before moving on to the next speaker. I think one of the questions that was raised that I was also thinking was quite interesting. So you talk about affecting stomatal density and conductance as a tool to improve drought resilience without affecting photosynthesis. And I think many people are thinking, well, how is that possible? Do you have any insight how we can change that without compromising photosynthesis and yield as such?
Yes. So changes need to be minor. So I would first say that this is because of the nonlinear relationship where we can move the needle from a high stomatal conductance to a lower point without disrupting photosynthesis much. So if we work in this area of the plot, we're likely to increase water use efficiency by a lot without major declines in photosynthesis. Plants tend to open the stomata more than they need. So this is why we have this plateau of photosynthesis with increases in stomatoconductance beyond a certain threshold. So the idea is to work beyond this threshold. Now, of course, there are some cases where the decrease in stomatoconductance are so big that leads to major declines in stomatoconductance beyond the point where photosynthesis is going to be stable. And this is when we start to have the negative impact in yield production. So the idea is to have minor to moderate declines in stomatal density such that we don't cross that threshold.
Okay, so I see. So there is some window in which we can work to really affect that.
Yes.
Okay. Then I think another question is, so this work is mostly done in monocot crops. So I don't know whether you can comment a little bit on how this translates to other species that we cultivate. Is this a kind of strategy we can apply?
So there are studying other plants. We have in here instances of tomato where they also decreased the density of the stomata and didn't seem to have any yield penalty. So it might be effective in other non-grass crops as well. But the majority of positive and successful cases are in monocots. So it might be that the structure of the stomata in grass stomata being more effective into regulating their aperture makes them more easy to be successful. So this is yet to be discovered.
Great. Okay, so I think if you like, you can continue answering the questions in the Q&A that have popped up. I see many people have interesting questions still, but we'll go ahead and go to the next speaker. Thank you again, Amanda, for a very interesting talk. So now it's my pleasure to introduce our second speaker of today, which is Professor Thomas Williams. Thomas studies ecophysiology from a more, I would say, systems biology perspective in combining metabolomics with systems biology. He was trained originally as a biochemist. He completed his PhD in plant sciences at the University of Oxford under the supervision of George Radcliffe and Lee Sweetlove, where he worked on metabolic fluxes within primary plant metabolism. Since 2012, he has held a professorship at the University of Brasilia in Brazil, where his group adopts a very integrative approach to understand how plant metabolism responds to changing environments. He combines experimental metabolomics with modeling, and he aims to describe with his research and to predict how metabolic networks reorganize under environmental stress. So today, Thomas will introduce us to his talk on genome-scale metabolic models in plant stress physiology, implications for future climate resilience. He will explain how genome-scale models, which is a type of constraints-based modeling, can be of use in the field of physiology. So go ahead, Thomas, you have the floor.
Thanks very much, Thomas, for the introduction. The audio is okay?
It's perfect. Thank you.
Perfect. Excellent. Okay. Well, so I'd like to start by thanking Plant Physiology for the invitation to participate in this webinar, and also to the editors of the Focus collection who've done an awful lot of work to bring together all these really interesting articles. So what I want to do today is to talk a bit about how we can construct and use genome-scale metabolic models to predict how plants may react to different environmental stresses. And what I'm going to do then is to pull out a specific example from our group that we cited within our review article to sort of illustrate a concrete example of how we can use these kind of models. So plants growing in both natural and in agriculture environments are always exposed to adverse conditions, and they've evolved a multitude of mechanisms to resist and tolerate these different situations. However, our predictions about the future climate point to increases in a range of different factors that cause stress in plants, Those include biotic stresses such as attack by pathogens or herbivores, as well as abiotic stresses such as higher temperatures, greater variation in temperature, flooding, and drought. All of these different types of stress have severe impacts on plant growth and survival, as well as agricultural productivity. So part of the impacts of these different stresses result from alterations that occur in metabolism. And I'm going to take drought as an example to explain a little bit about that. So when plants are subjected to drought, one of the things that they do, as Amanda was saying, is that they tend to close their stomata to conserve water. That generally leads to decreased photosynthesis as the rate of CO2 entering the leaf reduces. And it also results in increases in photorespiration due to increased leaf temperature and decreased CO2 concentrations. Under drought conditions, plants also tend to accumulate osmolytes, and they'll produce a bunch of different antioxidants, both enzymatic and non-enzymatic, in order to protect themselves from the reactive oxygen species that accumulate during drought. So what we can see is that even in this relatively simple example, we've got a whole bunch of different metabolic processes being affected, from the Calvin cycle occurring within the chloroplast, to the TCA cycle in the mitochondria, and to antioxidant metabolism occurring partly in the peroxisomes. Well, that means that if we're going to try and understand what's happening to metabolism under stress, we can't just focus on a single part of metabolism. We need to take a network view. We need to look at the whole of the metabolic network. And if we can do that, we'll have a better chance of understanding how plants actually respond to these different abiotic stresses. And we may also be able to engineer or breed crops that have greater resistance to the climates that we're predicting for the future. So what tools do we have available then that allow us to investigate the metabolic network as a whole? Well, one of the tools that we've got are genome scale metabolic models. So what's a genome scale metabolic model? It's essentially a representation of all of the metabolic reactions that exist within an organism, including all of the metabolites, all of the enzymes, and the genes that code for the synthesis of those enzymes. There's a number of ways that we can construct them, and I've tried to illustrate one of the routes you can take to do that on the left-hand side here. So typically we're going to start with a genome sequence that will then be annotated in order to assign function to all of the different genes that have been identified. Within those assigned genes, we're going to pull out all of the genes that code for enzymes and associate those enzymes with the reactions that they catalyze using different databases. Once we've got those enzymes together, we can make predictions about which metabolic pathways are actually present within the organism that we're working on. And then we can, from that list of pathways, we can assemble and curate a metabolic model. That metabolic model initially will be quite rough. And so you've got to do lots of curation in order to add reactions that are missing, remove reactions that shouldn't be present, and typically to also correct stoichiometry of reactions that isn't correct. But at the end of all of this process, we've got a large-scale model that accurately represents the metabolic capacity of the organism that we're interested in. These models don't have to be generic for a particular organism. We can do things in order to customize them and make them represent specific organs or specific cell types. So, for example, there have been lots of nice work done producing models of guard cell metabolism. So in that case, what you can do is you can restrict the reactions that are in the model to those that are specifically expressed within the guard cell. Or you can do the same thing, for example, for a leaf palisade cell. We can also couple models together to represent what happens within different structures within an organ, for example, within the structures that we might find within a developing seed, or we could even do that for an entire plant. So we construct multiple submodels representing different organs within a plant, and then we connect them with transport reactions that reflect movement of metabolites and inorganic substances between these different organs to create a whole plant model. What's more, we're not just restricted to spatial differences. As we discuss in the review article, we can also look at what happens over time, both short term, so we can build models that simultaneously reflect metabolism during the day and night, and that can be used to capture the processes of accumulation of photosynthate during the day and its remobilization to supply respiration during the night. And we can also look at more long-term processes. For example, what happens during reserve mobilization, during development of seedlings, or even through the entire life cycle of a plant. And there have been some really nice studies doing that, for example, with Arabidopsis and with rice. So we've got this possibility as well of coupling models together to reflect different times. Once we've got a model, that isn't the end of the story. What we've got to do next is to use this model to predict exactly how the network functions in the particular metabolic context that we're interested in. So we start with our initial metabolic model. And then what we're going to do is we're going to introduce a whole bunch of different constraints onto that model in order to force the model to operate within something which is within a range which is physiologically realistic. So we might go to the lab and measure the composition of a particular organ or cell type within the lab in order to derive these experimental constraints on how metabolism should be operating within the model. Once we've applied these constraints, we then need to use an optimization procedure. And that might be, for example, minimization of the total flux through the metabolic network, or in other words, the minimization of the sum of the velocities of all of the reactions. Or it might be, for example, maximization of some kind of efficiency parameter, for example, the lowest amount of substrate necessary to produce a certain amount of plant biomass. And once we've done that, we'll have a prediction of the way that the metabolic network is operating in the context that we've defined using our constraints and the structure of the model. This kind of work has now been done for a whole range of different crop and model plants. So we've got models constructed for a bunch of crops now, including, for example, rice, soybean, and corn. We've got model plants, for example, tobacco and Arabidopsis represented. And these different models created for these different plant species have been used to investigate a wide range now of different abiotic stress conditions that are associated with climate change. So we've got plenty of studies looking at increased and decreased temperature. We've got studies looking at the impact of greater photorespiration, which is often caused by greater temperature and alterations in CO2 concentration. We've got studies that look specifically at the effect of CO2 on metabolism. And we've got a couple of other stresses, for example, salt and drought being reflected in the literature as well. But what we've noticed when we were preparing our contribution to the focus issue is that there are actually plenty of gaps and plenty of opportunities for more research. So currently there are very few species that have models constructed for plants that are for tree species and very few for native plant species. It's basically focused on crops and a few model species. And also, there are a bunch of different stresses that really haven't been explored in as much detail as perhaps they deserve. So there's some nice studies that have been published on flooding and on salt in rice, but they're quite restricted. We don't have much information using these genome scale models about these stresses. So these are really nice opportunities for future research in this field. So what I want to do now, just for the last few slides, is to take out one example, which is an example that came from our group, which was investigating how soybean reserve metabolism is affected by drought stress, and use that to try and show exactly what kind of data we can generate using these kind of models. So I'm going to be talking just a little bit about the effects of effects of drought on metabolism during soybean seed filling. To try and address basically two questions, the first of which is the fact that the composition of a seed is very different to the composition of vegetative tissues in plants. So what are the metabolic processes that are used to efficiently convert substrate coming from the mother plant into the reserves that we're going to find within the seed? And secondly, this is something that Amanda also mentioned, seed filling is extremely sensitive to drought stress. So I'd like to explain and talk a little bit about how the metabolic network is affected in developing soybean seeds. So to do that work, we had to go outside the lab, and we joined a nice field experiment that's run by some colleagues at Embrapa Cerrados here in Brasilia. And they've got a really lovely experimental setup where, because of our very long dry season, they can do drought experiments under field conditions. So in this setup here, an irrigation bar passes along these rows here, and it applies different amounts of water to different rows. So these plants on the left have received plenty of water, whereas these on the right are obviously very unhappy and they've received very, very little. So what we did is we joined in with this experiment and we harvested plants beginning at R5.1, so the beginning of soybean seed filling. And that was at 72, 82, and 97 days after planting. We collected the developing seeds, we took them into the lab, and we measured the composition of those seeds to try and see how drought impacts the process of seed filling. And what we noticed was that decreased irrigation led to much earlier seed reserve accumulation. So the plants are producing fewer seeds, smaller seeds, but the process of reserve accumulation is happening faster, right? So what we can see here is that with this process of stress happening, we get a greater increase in fatty acids, which is shown here under the conditions of stress, and a decrease in the amount of starch that's present at this last harvest point here, indicating that the seeds have moved further through the process of seed filling. So with all of that experimental data, the mass of the seeds and their composition, we can derive those constraints on our metabolic model that I was talking about earlier, plug those constraints into the model, use our optimization procedure then to predict the metabolic fluxes that are happening in 8 different scenarios, which reflect 4 different levels of irrigation and 2 different time periods. So what exactly does that show us? I've taken just a little corner of the metabolic network to show to you today. What we see is that the model predicts several things that are common to all of the different scenarios we tested. So you can see in this metabolic map, there's a lot of carbon that's coming through glycolysis here that's being directed to the synthesis of fatty acids within plastid, which makes sense. Soybean accumulates large amounts of oil. There's not really very much TCA cycle flux. And the TCA cycle isn't using pyruvate as a substrate. This reaction basically isn't operating. It's using glutamine, which is coming from the mother plant as well. And this all makes sense. Soybean seeds are green, so they have photosynthetic activity. That means that they're producing lots of ATP and NADPH in the plastid, in the chloroplasts. So there's less demand to use mitochondrial respiration to produce energy and reducing equivalents. There's also the opportunity to shuttle reducing equivalents into the mitochondria to contribute towards ATP production. So the results show an interesting pattern in terms of flux predictions under the different levels of irrigation and the 2 different time periods we investigated. During early seed filling, increased stress, or in other words, less water, resulted in a tendency to have increased fluxes through these reactions. Whereas during the second period of seed filling, so later during seed filling, we had a decreased flux in these different reactions as we went to higher and higher stress. So we've got qualitative similarities, but also quantitative differences. So what exactly is going on here? Well, to try and understand that, what we did is we expressed all of the fluxes relative to the amount of carbon that was coming into the developing seeds. When we did that, we constructed these heat maps, and you can see quite clearly here that for the first part of seed filling, which is represented here, we don't actually have much impact of drought on the relative fluxes through the different reactions within our metabolic network. What that indicates is that a decent amount of the impact on flux that we were seeing, the impact on the network, is actually due to the reduced amount of photosynthate that the mother plant is able to supply to the seed under drought. On the other hand, during later seed filling, which is represented here, we've got a whole bunch of different reactions that are perturbed under greater conditions of stress. So, for example, we've got these increases here in the activity of reactions associated with chloroplast photosynthesis within the chloroplast, and with fatty acid synthesis as well, whilst we've got decreases in reactions associated with the mitochondria. So the fact that we saw photosynthesis being flagged in this previous analysis made us think, well, we've got a model. We can see what happens if we alter the amount of light that's available to these developing seeds. And that's one of the really nice things that we can do with this kind of modeling. We can test what happens when we manipulate different parameters. So what we did was then to alter the amount of light that's arriving at the developing seeds and look at the effect that that has on carbon conversion efficiency, so the amount of carbon coming into the seed that then goes into the reserves, and also the underlying fluxes that were occurring within the metabolic network. What you can see here is that as we increase the amount of light, so going from left to right here, we get a gradual increase in carbon conversion efficiency, which makes sense because we've got more energy producing or converted into ATP and reducing power by the chloroplast, so less need for mitochondrial respiration. And that increases quite gradually, but we've got these rather more sudden shifts in the activity of these other reactions. So what that's showing is that the most efficient way to convert substrate into seed reserve varies depending on the amount of light that is available. And the other thing that was quite nice here is that our model was able to predict a particular way of operation of the Calvin cycle, known as the Rubisco shunt, which is where we've got activation of Rubisco, so flux through the reaction that fixes CO2, but without activity of the rest of the Calvin cycle. So you can see that this SBPase reaction here is essentially zero. And that's a particular metabolic shunt that was described quite a while ago now, and has also been shown to occur in developing soybean seeds using carbon-13 labeling experiments. So just to wrap up, I'd like to reinforce that we can use metabolic modeling then to help us understand how plants respond to the different stresses that are caused by climate change. There are studies for a range of stress conditions in model and crop plants, but non-crop plants, trees, and certain stresses like salt and flooding have been underrepresented so far. I tried to show a little bit about how the effects of drought, or the effects of drought on soybean seed metabolism vary between early and late seed filling. So early on, it was more a question of substrate supply, whereas later on, there was more of a rewiring of the metabolic network. And we were able to show that seed photosynthesis is quite important, especially under stress conditions. So that might be an interesting target for metabolic engineering and breeding. I'd just like to finish by thanking the people that contributed to the work. Erica, who wrote a large chunk of our review that was contributed to the focus collection and did the metabolic modeling work in the other study. Christina, who did a huge amount of work in the field and in the lab together with our undergraduate students, and Walter and Lucrecia, who have this wonderful this field experiment system that they allowed us to participate in, and also to our funding agencies, FabDF and CNPq. So thank you very much for your attention. I'll be very happy to answer any questions.
Thank you very much, Thomas. It was quite an interesting talk, a bit far from my own research, but I think what you presented is a very powerful method with both predictive power and a power to test hypotheses that you don't need to test in the field, but you can first test in computation, through computational approaches. I like it very much. Since we're running a bit short on time, I see that there is a lot of interesting questions in the Q&A. So I would propose that we head on to the next speaker, because otherwise we might run out. But feel free to answer all of the questions that have popped up in the Q&A section. I think people will be more than happy to hear your opinions. So let me introduce our third and final speaker, Dr. Sabina Viadangos from the University of Barcelona in Spain. Sabina performed her doctoral work under the supervision of Professor Sergi Munebos. Sabina is an ecophysiologist by heart, and her work focuses on understanding how stress-tolerant plants cope with extreme environments. She is particularly interested in photosynthetic acclimation, antioxidant regulation, and hormonal responses under these extreme environmental conditions. She currently continues her research in Barcelona, where she's developing new research lines on the climate effects of both terrestrial and marine species, but she's also translating her findings into more applied and biotechnological context. The talk she has prepared for us today is based in part on her doctoral research. She will share some exciting things on her work on the plant Sempervivum tectorum, which is a drought-tolerant succulent species with an incredible capacity to withstand extreme environmental conditions. So, Sabina, it's your turn. The floor is yours.
Okay, you see my presentation? Yes.
Yes, it's not on full screen yet for me, but...
Okay. Okay, so thank you, Thomas, for the kind introduction. And I would like also to thank the American Society of Plant Biologists for the invitation to participate in this webinar. So I'm going to explain to you the key like strategy that underlies this extreme stress tolerance in houseleek. So first of all, as aerobic organisms, plants are prone to oxidative stress when meeting their metabolic and developmental demands. And in chloroplast, the excess energy from photosynthesis can lead to the inevitable production of reactive oxygen species. So under stress, increased ROS can damage the photosynthetic machinery and cause oxidative modification in lipids and other cellular components in cells that can trigger this photosynthetic stress and lipid peroxidation in thylakoid membranes. The first product of lipid peroxidation is the lipid hydroperoxidase, and then the second product is the malondialdehyde. Malondialdehyde, or MDA, is a product that can be highly toxic to cells. Even so, plants have evolved efficient mitigation strategies, including the antioxidant machinery and phytohormones that can reestablish the redox homeostasis in the cell. So, moreover, when these ROS are produced transiently, they can act as signaling molecules in the plant stress alarm system, and together with oxylipins, the products generated by lipid peroxidation, can perform retrograde signaling from the chloroplast to the nucleus to activate stress response genes, and then enabling acclimation to environmental stresses. Because plants grow in a modular way, they can constantly replace these damaged parts, such as leaves and roots, and this allows them to compartmentalize damage, discarding damaged organs and resprouting new ones after environmental stresses. So this can help some plants, especially perennials, to avoid senescence in the whole plant and live for a very long time. Here we can show two photos of the Cistus albidus, which is a Mediterranean drought-tolerant shrub that shows a clear recovery after rainfall following a stress period. But however, this extreme stress tolerance may come at a cost. And in fact, extreme stress conditions trigger a fundamental trade-off between growth and survival, with enhanced stress tolerance often coming at the expense of plant productivity and vigor. While survival is secure here, this competitive advantage given by growth is reduced, and then it establishes this trade-off. In this trade-off, oxidative stress plays a regulatory role among balancing this vegetative growth, this plant defense, and also the reproduction in plants, leading to survival in stressful conditions. Among extreme survival strategies, stress-induced quiescence is a reversible slowdown of growth and metabolism, and is primarily related to meristem, but it also is being observed related to cells and active differentiated organs. Among the adaptive strategies related to prolonged droughts, there are different adaptive strategies that have evolved within the plant kingdom. One strategy known is the dehydration tolerance that is illustrated by these resurrector plants, and is the ability to withstand near complete loss of water in their tissues and recover the metabolism upon rehydration. But in contrast to this strategy, there is another one that is the dehydration avoidance that is depicted by this Crassulaceae and Cactaceae family, and that refers to the ability of plants to maintain the hydration in tissues and despite this limited water availability. This strategy is mainly employed by succulents, and specifically the Crassulaceae family have all cell succulents in leaves in which all or most cells are both photosynthetic and capable of storing water in large, highly vacuolated cells. So this leaf surface anatomy is linked to the aridity and the water harvesting strategies. However, maintaining cellular function despite reduced leaf hydration is also considered a drought-tolerant strategy, and where the plant internally is capable of osmotically adjust the water volume without compromising the proportion of other cellular components. Plants with clonal growth face an additional energetic challenge, because clonal growth allows an individual to produce a genetically identical but potentially physical and physiologically independent offset, but they are often supported by maternal resources, at least until they establish into the root. So under these stressful conditions, and if the division of labor it doesn't fail, so it doesn't occur well, or maybe the unrooted offsets becomes too costly for the mother, clonal plants may stop supporting dependent ramets. So now I'm going to present you the main character here, which is Semperbium tectorum. It's also known as houseleek, and it's a clonal plant that is native to the highest European mountains. This Crassulaceae species is pluriannual with evergreen leaf rosettes, and it propagates vegetatively through stolons, so that the parental rosette is producing several offsets that remain attached until rooting into the soil. Each rosette is monocarpic, growing for 3 and 4 years until it flowers and fruits only once before the leaf senesces and the rosette dies. So the Semperbium genus is well known for its extreme tolerance to abiotic stresses, but its tolerance to prolonged draw stress and underlying mechanism remain unknown. So here, the aim of this paper was to elucidate survival strategies to withstand an extreme draw stress in this clonal plant species at various organizational levels, including parental and offset rosettes, with a particular emphasis on the oxidative stress homeostasis control. First, we observed that unrooted Semperbium tectorum rosettes could survive without any substrate or supplementary water for a whole year, showing outstanding desiccation tolerance. The rosette folder the innermost part, hence protecting the youngest leaves and the apical bud of the rosette from desiccation. When we expose Semperbium tectorum potted plants to draw this show that this species can withstand very severe stress, because it after 7 months of nutrient solution withdrawal, offsets were still alive, and the parental rosettes recovered their photosynthetic and water statuses only 1 week after the recovery, after rewatering. Indeed, a tetrasodium viability test indicated that unrooted offsets maintain the highest, the lessest leaf damage and the highest viability, in fact, than the parental rosettes and the rooted offsets. And these results indicate that this clonal species efficiently protects meristematic tissues in the apical bud to ensure regrowth, as we can see here with the green area measurements that indicate this gradual senescence and recovery after rewatering. So to investigate the physiological mechanisms underlying this extreme stress tolerance, we examine the hormonal and antioxidant contents in the middle leaves of the parental rosettes. We saw that the abscisic acid, the stress-related abscisic acid peaked during the moderate stress, and also this draw stress altered the jasmonase metabolism through an accumulation, until an accumulation of jasmonase solution, which is the bioactive form. Regarding photosynthetic stress, we found that the malondialdehyde, this toxic secondary product from peroxidation, was lower in the stress than in the recovery and in the previous moment. And we saw a photoprotective strategy in which we saw an increase from the chloroplast lipophilic antioxidant vitamin E, specifically from the most active form, which is the alpha tocopherol form. Overall, these results suggest a Kirsten-like strategy in the middle leaves to maintain low oxidative levels during prolonged periods of stress. So then, prompted by these results obtained under control conditions, we then investigate the draw acclimation response of this clonal plant in its natural habitat. We selected from the southern latitudes of native range 3 populations, and across a draw and altitude gradients to study their seasonal course. So we study from the spring to summer and to the autumn recovery. And we see here the microclimatic conditions of these 3 populations, and in population 1 was the most affected by the summer draw. Although population 1 was the most affected by this summer draw, the relative water content only decreased until 70% in the middle leaves of the parental rosettes, and the slight photoinhibition also recovered. Population 2 was also this experienced these mild reductions, but this sensitiveness was less in a lesser extent, and population 3, which was the highest altitude located, did not experience any variations. Despite senescence of the outer leaves, the parental rosettes kept also some unrooted offsets alive for several months. So this showed that this clonal offspring was a priority in stressed plants under natural condition also. But maintaining the parental rosette and clonal offsets carries a physiological cost. And indeed, we found adjustments in clonal offset production that was this variable was influenced by the season. So we saw that in the growing season, we saw an increase in the production of new clonal offsets, but it was reduced in the autumn due to the summer. And also the population 1 that was the most drought affected has a less extent of clonal production compared with the population 3. So also when we found, when we checked for the stolon, this clonal growth organ, we saw that in population 1, that was the most drought affected, we found an increase at higher levels of abscisic acid and lower contents of the bioactive cytokinins related to growth. Then, among all the measured functional stress markers in leaves, we perform a PCA comparing all the data from the 3 population and the 3 developmental status of the roses, parental, rooted, and unrooted. And we found that PC1 component, the first component, was related to plant oxidative status, and the second to hormonal plant stress responses. Focusing on the oxidative status, we also found in the file a significant decrease in lipid hydroperoxide content, which means that these plants exhibit low oxidative levels and an increase in alpha-tocopherol. When we checked for the hormonal responses, we also found 2 simultaneous peaks from abscisic acid and jasmonylileucin, and we also found an improved hormonal and photoprotective mechanism in offsets, displayed by higher levels of gamma-tocopherol, which is the precursor of the alpha-tocopherol, in the offsets. And also jasmonylileucin was higher in the offsets than in its parents, rosettes. So finally, we want to investigate the costs, if any, of the extent of clonal reproduction in the drought acclimation response of this species. And from all the individuals included in the study, we selected the ones that clearly show Kiersten metabolism under stress. So among these individuals, we found that neither the number of unrooted offsets nor the total clones it affected the stress survival of the mother rosette as displayed here with the stress markers. But however, in response to an increase only in the number of unrooted ramets, the parental rosettes increased the abscisic acid contents and also the photoprotective capacity and leaf mass area, which is a morphological adjustment. So in summary, as a summary of this study, Semperbium tectorum is a highly tolerant drought species that is endowed with both morphological and physiological strategies, and they are spatiotemporally displayed to protect the meristematic tissues in the inner apical bud. Being tolerant to severe stress allows clonal offspring maintenance attached to the parental plant during prolonged periods of time until routine occurs. And moreover, this Kiersten-like strategy displayed by the parental rosette is modulated by abscisic acid, by photoprotective and leaf mass area adjustments when the number of unrooted offset increases, but at no cost for stress survival. So by prioritizing the survival of the clonal offsets, the whole gene survive to a drought stress, even when the parental rosette senesces and dies, it can reach the inherent immortality for the genotype. So this species is adapted to thrive in harsh environments, not only by a clonal, but also sexual manner, reproduction, and overall our findings support an unusual change in oxidative stress homeostasis in leaves that is achieved via downregulation of the metabolism to a Kiersten-like state under extreme stress conditions. So finally, I want to acknowledge all my Plant Synergy research group members, and which is led by the Professor Xinguang Zhu and the funding institutions. And if you have any questions, I would like to answer them. Thank you.
Thank you very much, Sabina. It was a very interesting talk. I think for many people who are not working with these extreme specialists, these things are very interesting to see and to learn what plants can actually do in these environments. I see that we're actually already over time. We're 5 minutes past 4. I think maybe I have one last question to kind of finish the webinar, which is basically you work with two different kind of settings. First, a setting in the lab, and then later in the field. And I think for most ecophysiology studies, this is kind of a tricky point. No, so how do we translate these kind of things? So I don't know whether you can comment briefly on this.
Yes, Thomas, I think that this is the classic challenge in Ecophys to compare the greenhouse experiments to the field, the real life ones. But I think that both of them complement, and to confirm the mechanisms found in the greenhouse control experiments also in the file is needed to have the whole picture of this plant, like its whole response and its real response in nature conditions. And also we can like compare the stress intensity by stress markers. For instance, relative water contents of 70%. They got this stress, no, in the moderate in the greenhouse and also in the file. So with these things and to quantify microclimatic conditions also, it's good.
Yeah, so it's like a multi-strategy to control like whether your experimental setup can be translated. Okay, so given that we're at the end of the webinar, I think I speak for most speakers. If there's any questions that the listeners are still having, they can always contact you guys to further ask and talk. And with this, I would already, from my part, like to thank the speakers and the organizers for a very interesting webinar, and for sure also the listeners of today. Thank you very much for being here. I will give the last word to Mary Williams.
Thank you. Thank you, everyone. I actually learned a ton. I thought it would be way over my head, but I was really engaged. And really, Augustin, on behalf of all of us, please send our regards to the rest of the editors. This is such a fantastic collection of papers. You guys have done a great, great job. And again, speakers, thank you for your time. Listeners, and Thomas, Augustin, Sabina, Thomas, and Amanda, thank you so much. Really appreciate your time.
Thank you. Thanks, everyone.